All right, guys, in this video, we're going to start looking at the cell cycle and namely cell division. All right, so let me get a couple things squared away here on my screen. Get my pen ready. Here we go. So some questions we want to be able to answer. Number one, what is the cell cycle? All right. What is the purpose of mitosis? And then we want to know each step, all right, in mitosis and what happens in each one. All right, so we want to be able to describe those steps. So some things that we already know, right? Number one, from cell theory, cells come from pre-existing cells. And we looked at some of this in the previous lessons, but we know that, human beings start off as a fertilized egg, right, called a zygote, which grows and divides and becomes all the different cells in your body. It goes through specialization, right, and we looked at all that before. All the cells in your body, since they start with that one cell, have the same set of genes, okay? And the problem with making big organisms from these, singular cells is that there is a size limit, all right, on how big a cell can get. Once it starts to grow and grow and grow and grow, its size actually starts to become a hindrance to its ability to continue to be alive, all right? It can kill it. The cell can die, right? The size can kill it if it gets too big. So the reason behind this, the scientific definition that we talk about, is the ratio between surface area and volume. So the bigger something gets, all right, the smaller the surface area to volume ratio becomes. That means there's less membrane that is available for diffusion and osmosis. So, I've got to get more and more and more material in to keep the cell alive, but the membrane's not that big. It doesn't get, exponentially bigger. So as the cell gets bigger, that poses a problem. The cell cycle is the answer to that problem. So there's two main sides of the cell cycle. There's interphase and there's cell division. Now, the majority of your cell's life they spend in interphase because your cell has a job to do, right? If it's a liver cell, it's got liver stuff to do. If it's a red blood cell, it's got red blood cell stuff to do, right? If it's a brain cell or a neuron, it's got neuron stuff to do. And if it doesn't do those jobs, then you as an organism will not function correctly. So the majority of a cell's life is spent in interphase. But when the cell gets ready to divide, it's time to jump over into cell division. And you can control cell division, all right, by the amount of nutrition you give a cell, by different chemical signals you give a cell. There's a lot of different ways we can control cell division. Now, to kind of zoom in on interphase, there are three main parts of interphase. This is the working phase. And those three subparts are G1, S, and G2. G1 is when the cell is growing. So it's just been, it's a daughter cell, right? It's just come from the split of two other cells. So it's small. It's got to grow. All right, so it's bringing in nutrients. It's building up the cell machinery that it needs to function. It's growing. Once it gets through the growth phase and it's mature, it's the right size, then the DNA is going to replicate, all right? And it's going to replicate because when the cell divides, it's got to give a full copy of the DNA to each of the daughter cells. And then after the DNA replicates in S phase, you have the G2 phase. This is a second growth phase. It gets just a little bit bigger, does a little bit more work as far as its job is concerned. Now, in order to talk about this S phase, we got to talk a little bit about chromosomes. So when we look through the microscope or we look at a cell, sometimes we see chromosomes, and they're these really thick structures that are basically comprised of condensed DNA and some proteins. The proteins are called histones. When interphase is over, you're going to have the chromosomes duplicated, all right? You're going to get, a singular full copy for each daughter cell. And we're going to talk about those in terms of chromatids and the centromere. So when you think of a chromosome, you probably think of something that looks like an X, right? That's usually how we draw chromosomes. What this actually is two chromatids, and they're connected by a centromere. That's a cool word. Centro means, the center or the middle. And mere comes from the same root as mer, which means part. So this really scientific name for the unit that joins two chromatids is called the middle part. That's the centromere. The chromatids, there is a left chromatid and a right chromatid. They're called sister chromatids. But here it is. We have sister chromatid number 1 right there. So that's a chromatid. And then we have sister chromatid number 2. And you could do left and right, it doesn't really matter. But the thing that we want to know is that the left side and the right side, it's one long strand. They're called chromatids. Now, this is what a chromosome looks like just before it divides. Cell division is going to take place in 2 steps or 2 processes. If you're just dividing your genetic material in order to make 2 daughter cells that are clones of the parent, then you're undergoing mitosis. If you're making gametes, which is the scientific name for a sex cell, like a sperm or an egg, then you're also going to undergo meiosis. Cytokinesis is the last step in both of these, and it's the movement of the cell and the cell contents in order to form 2 distinct daughter cells. Remember that cyto, that prefix, means cell, and kinesis means movement. All right? Cytokinesis, cell movement. Cyto can also be a prefix for cytoplasm. Can mean that sometimes. So you could also think of this as the cytoplasm is moving apart. Now, mitosis always results in 2 daughter cells. They're identical to the original cell. This means that they should be clones. You want to know this whole little sentence here. This is really important. There are 4 steps in mitosis. We have prophase, pro meaning before, right? The first. Meta, talking about the middle, which we'll see in a moment. And then we have ana and telophase. We use the acronym PMAT, PMAT, to remember this. Now, P is for prepare, right? Pro means first or before. The prophase step is the first step of mitosis. And in this step, your chromosomes are going to coil up, your nucleus is going to disappear, and in animal cells, we're going to find these spindles. Now, these spindles actually appear in both animals and plant. The centrioles will only appear in animals. Now, I've only drawn a couple things in this diagram here, this cell diagram. You'll notice I haven't included any of the organelles. We're just focusing on certain things. So, number 1, this is a dotted line for a reason. This is the nuclear, right, envelope or nuclear, we also call it the nuclear membrane. And that nuclear membrane is going to break down. So the chromosomes are going to be just out in the cytoplasm. These are our spindle fibers. And that right there would be a centriole. Now, those were kind of hard to draw in PowerPoint, but they're actually these like column or these spherical. There's like that sphere right there. They make these like spheroid kind of microtubule columns. It's pretty cool now. They usually lay side by side each other, kind of like that, which is interesting, right? In metaphase, M is for middle, right? Meta means the middle. All the chromosomes are lining up in the middle of the cell. And the spindle fibers are going to attach to the sister chromatids at the centromere, right there. That's metaphase. The chromosomes are in the middle. In anaphase, you remember A is for apart. The sister chromatids get pulled apart. So this chromosome is made of 2 parts. They are getting pulled to the 2 opposite sides of the cell, right? Just like that. And in telophase, the prefix telo means different, just like telephone to call someone from a distance. That's where we get the word tele. Telo is a Greek prefix. It means distant or far or apart. And you can remember that telophase pulls apart the cell. I'm getting new nuclear membranes, so they're reforming. The chromosomes are uncoiling, right? The spindles are gone. The spindle fibers, they're gone. The centrioles are gone, and cytokinesis starts. Now, cytokinesis is the movement right there. It's this pinching of the cell. And eventually, you're going to get 2 new cells, right? Cell number 1 and cell number 2, just like that. They'll have 2 daughter cells. Now, during cytokinesis, we have a little bit of a distinguishing difference between animals and plant cells. In animal cells, the membrane just pinches off. It's just done. It's just here, pulls apart. In a plant cell, you get a cell plate, which eventually becomes the cell wall. So if I was looking at this as far as I say, let's make a bigger plant cell. So here's my plant cell, right? We're going to draw it as a rectangle because, you know, it has a cell wall. Then in the middle, where cytokinesis would be occurring, this would be called the cell plate. And that's eventually going to become a new cell wall. Remember, plant cells have a cell membrane. It's right inside the cell wall. So that's also going to be pinching off. But the cell plate does differentiate plant division from animal division. So in this PowerPoint, that's it. Right? We want to know the cell cycle. So the cell cycle has two parts. It has interphase. So let's write this out. I need my pen to work here. Come on, there we go. Interphase. Oh, there we go. Interphase. And then we have cell division. Those two L's look kind of rough. There we go. We have two parts, interphase, cell division. The purpose of mitosis is to create two cloned daughter cells. The steps of mitosis, we can remember those with PMAT, right? Prophase, metaphase, anaphase, telophase, all right? The steps, as far as what happens, that's in the PowerPoint. All right, make sure you know those steps, right? That's going to be important moving forward, okay? So that's basics of cell cycle, all right? Something I'd like to point out, I'm not sure if it's in the next PowerPoint or not, but the cell cycle is a cycle. So PMAT, right? If we look at this kind of like a clock. So this would be CD, cell division. We have G1, S, G2. So from here to there, all that is interphase, right? Interphase, going around there. So you can see that most of the cell's life is spent in interphase. That's important to know. All right. See you guys in the next video. If you have questions, let me know.